So things that are bigger settle faster. So it turns out that settling velocity goes like diameter squared if you're in very small, in the Stokes regime, and still more than diameter to the first power. Juliana, you wanted to say something about size. Why do we care about size? Affect flocculation, so affect interaction between particles. Resuspension is size dependent, again, because things that are bigger settle faster.
Ecology.
So, ecology, in what sense?
Community composition.
Community composition, size is something that describes the community composition. Do we have bigger cells? Do we have smaller cells? They tend to be specific group that are associated with the bigger one, like diatom, as we're told. Smallest one tend to be what we call picoplankton, like cyanobacteria or prochlorococcus, prokaryotic organism as opposed to eukaryotic organism. Josh? That's it. Oh. What? Grazing. There's more food in a big cell than in a small cell. So the food chain passes up material differently if it's a few big cells versus a lot of tiny cells. So some of the reason why we care about size. And maybe a few optical oceanographers care about size because it's affecting the optical property, and vice versa. We can use optical property to say something about size. Okay, so then we move to the next problem, which is, we talked a little bit about processing. What's the size of something like this? I just said we want to know about size.
100 microns?
No, that's 100 micron, but is 100 micron this size?
No.
Is it this size? Is it this size?
It depends which species because Serratio.
Yeah, there's many different Serratio. But it's really hard. We try to quantify size. What's my size? Is it my height? Is it my width? Or this direction, this dimension? When I take it, my gut in. So no, but it's a major issue. Is so people have come up with all kind of ways to do it. It might be the diameter of a sphere that has the same volume as I do. So that's to be something probably pretty big. It's going to be something like this. That would be the sphere that's going to be the same volume as I am. Or the sphere that has the same average cross-sectional area over all orientation. So, sometimes I'm skinny in this direction, big in this direction, and all other possible directions. So we're trying to describe something very complicated that can interact with light in very complicated ways by one number that we call size. So we already know it's an approximation. And this is something you have to remain in mind. And then, okay, so maybe we can come up with a long axis and a short axis. Maybe we can come up with all kind of other ways. And a lot depends also on the methods that we use. So let's assume we figured out the size, and then we try to build a size distribution. So the way you build a size distribution is you say, okay, I have a bin. Let's see how many of my particles are in this bin. So it should be, I'll have 20 here. In my next bin, maybe I have 40 particles. And then in my next bin. And how I do the binning will affect the number of particles in my bin, or this might be 35. If I put a smaller bins, the numbers are smaller than if I have wider bins. Because if I have this wide, I'm going to have 75 particles. If I have only half of it, I have 35 in this one, 40 in this one. So how you build a size distribution is important. And it's important, therefore, to look at units and figure out how people did it. One way to move from a histogram like this into a continuous size distribution that we can fit functions to, etc., that are bin independent, is to divide by the bin side. So I'm going to look at the number in each bin, and I'm going to divide by the bin size. And this way, I'm going to stop being bin dependent. to some degree. I mean, it still depend on, the discretization depends on this, but I can have a big bin and a small bin, and I'm taking into account the fact that the bins are different sizes. And you can also, so this will be a frequency diagram of how things are distributed, and then you can also, you'll often see people that look at cumulative diagrams of size distribution, because this is really a probability of being between one size and another size. If we don't put numbers here, just frequencies. We can multiply by the total number, and that will give us the numbers we have in each bin. So far, so good? Okay. So as we move from discrete, we have to deal with bin side, we said, and that's what we call differential size distribution. The size distribution where we divide by the bin. What do you think will be the units of such size distribution? What?
Per meter.
So you'll have just per meter. I'll have
Counts per meter.
number per meter. So I have number per meter. No concentration. Does it matter if I do it in a meter cube or centimeter cube? Will I get more numbers in a meter cube?
You can do it by the surface area of the particle or by the volume of.
So no, first we talked about having numbers in these bins. So we have numbers of particles. In this bin, the D, you're right, the D is based on whatever length scale we chose. An equivalent sphere of same cross-sectional area, same volume, same something. That's how we get to one number. But once we agreed on the type of size of diameter, so this is D1, this is D2, D3, this is the average in this each bin, we have numbers. But those are numbers per, here we have numbers per concentrations. No, so this is number in this bin, but it's a number per meter cube or centimeter cube, whatever I collected. So that's a concentration. So I have number, so in each bin here I have, in this one I have 20 per, if it was 1 liter, 20 per liter in this size. But as I move to a size distribution that's continuous, where I divide by the bin width, now I get another m to the minus 1, as Josh said. So it's going to be now number L to the fourth, per L to the fourth. Okay? Because I have the concentration per bin width.
We deal in concentrations, not in frequencies.
The difference in frequencies and concentration is that you're multiplying this by the total numbers of particles you have. Because you want to know, the shape doesn't matter. The shape of the frequency will be the same, but you want to eventually integrate to say how much material do I have, for example. How much carbon do I have? It's everything that's within those bins. So I want to add them up. Okay, so then what you'll find is that people will generate, usually there's 3 size distributions that you'll see generated. And they all describe the same data very differently. You'll have a number distribution. And when you look at numbers in the ocean, the most abundant organism, most abundant phytoplankton is Prochlorococcus. It's most abundant. But once you look at volume, it's not the one that has the most carbon of all phytoplankton. It's tends to be the intermediate ranges, the nanoplankton and the microplankton have more volume than the picoplankton. But suddenly you're looking at the same exact data. And look, it's emphasizing a completely different group than when you do a number distribution. And you can do also an area distribution. People do. And when you do that, when you compare a number distribution with a volume distribution, your mean size is different, because the mean size here is emphasizing the mean based on the volume. Similar to the mean height, the total surface area is the same in both, and the standard deviation around that is also going to vary. It's going to be different. And the other thing you'll find is that it's logarithmic scales we're using. And that goes back to what we talked before, but I'm going to add some more information about it in a second, why we're using logarithmic size bins. So when you look at, this is a great paper from Jackson, 1997, where he took a whole bunch of different sizing instruments and just bridged all of the different instruments such that he has a size distribution going from, if this is in centimeter, 10 to the minus 4 centimeter is 10 micron. So here it's about 10 micron. No, sorry, this is centimeter. millimeter, micron. Yes, so this is micron. This is 10 micron, 100 micron, a millimeter, and a centimeter here. And from going from cameras to other sizers, all in situ sizer, he was able to build this size distribution. So this is in numbers per centimeter to the -4. Then integrate to make a volume distribution. Same exact data. This is 30 meter, 15 meter, 10 meters in the water. It looks very, very different. Again, this is the volume distribution. And this one, he actually, in ppm, so part per million, ppm. So that's volume of material per volume of water. So here it's not normalized by the beam size. But suddenly it's emphasizing the big ones suddenly become the most important ones. So depending on how you present exactly the same data, you're going to emphasize completely different parts of the... So there's more volume in here than there is in here. So in terms of food, maybe this is more important. Depends what you're interested in. So as we talked yesterday, different size methods or sizing methods are sensitive to different sizes. And that's just because there's this resonant curve we talked about, and I'll show you some more. If you want to do a centimeter size organism, you're going to use acoustic. Optics is sensitive to small particles. If you're using imaging, that's different. So now depends on your imaging system, how many pixels, what's your smallest pixel. That will determine what you can resolve in terms of particle. And you'll see the IFCB today, and you can see certain. It's an imaging system, it's a microscope, and that's sensitive to its own things. When you use optics, you usually, if you're in the geometric optics limits, when you deal with particles significantly bigger than the wavelength, what are you sensitive to? Are you sensitive to their cross-sectional area, or are you sensitive to their volume? Cross-sectional area. In geometric optics, it's cross-sectional area. So what matters is the average cross-sectional area of particles with the optics in that regime. Okay? When you use something like Coulter counter, what are you sensitive to? Coulter counter is something that it passes particle in a fluid that's conductive, and it's looking at the changes in impedance. You're sensitive to? Coulter counter. Volume. Exactly. You're sensitive to the volume. So when you compare these two methods, again, you have to know something about cross-sectional area to volume. For spheres are an extreme shape. They're not an average shape. They're an extreme shape. They have the least average cross-sectional area per volume. Okay? So it's not a good average shape. Every other shape that's concave will have a bigger cross-sectional area to volume. Average cross-sectional area over all orientation as well to volume. So spheres are really an extreme, something that's important to remember when you're comparing method. And many sizing systems have problem in both ends. In the low one, it has to do with what's your threshold to even detect that there's a particle. How do I know there's a particle? How much fluorescence it needs to give me, or how much scattering it needs to give me so that I know it's there? Versus in the large ones, big particles are rare. What's the likelihood they're going to be in my sample? What's my counting uncertainty of a big particle in my sample? And if they can swim away from me as I collect them, the bigger they are. So this is a really classical paper every one of you should look at. Sheldon, 1972, addressing size distribution in the ocean. Basically what they see is that to a first approximation roughly equal, a hypothesis is presented to show that to a first approximation roughly equal concentration of material occur at all particle size within the range of 1 micron to 10 to the 6 micron from bacteria to whales. So basically, in logarithmic size distribution, as you go from 1 to 2, 2 to 4, 4 to 8, etc., or 1 to 10, 10 to 100, 100 to 1,000, you'd expect the same volume of material in each of these beans. That's a very strong assumption. Don't tell me it's dying. Good. It's not. And is it? I hope it is. Let's put it in the other side. Yesterday, that's what killed it. Okay, so this is really strong, but they use Coulter here. They looked all the way to Wales, and if you look at this scale, it's only a factor of 2. This scale, a factor of 3. Phytoplankton to tuna, whales to phytoplankton. 1972. And this is consistent with what you saw earlier today, a size distribution that goes like d to the minus 4. So what we call the Young size distribution is exactly this, one in which volumes are constant in logarithmically increasing size bins. It's really cool. And this is kind of the canonical. Of course, there are changes relative to it. But if your size distribution is really, really different from it, you may want to check again. And this is what Colin talked about also yesterday.
So the difference between the slope of the -3 and the slope of the -4 is the
Division by the bin size.
By that, but
Yeah.
then that also gives you an extra loss term of a factor of 10 if you look for volume, and that is the factor of 10 of growth efficiency. So the -4 is actually really ecological because it actually, it's not just the conservation of volume through the ecosystem, it's the conservation of volume or mass with an inefficiency, which is what we expect. So.
And there's a lot of paper trying to explain this incredible observation. I mean, with all kind of allometric relationship, there's a lot of work that's being done to try and explain it. But this is really the first paper to address it all over all these scales. Okay, so you saw that. So this is one power law type of power law size distribution. It's the one that's power law that you saw also from Kurt. But it's not the only one people have used to apply to ocean particles. You'll find those like this gamma distribution, and this is from a paper by Bricot and Morel, and those tend to apply really well to a group. You want to do a specific phytoplankton, and this was fit to a culture. It will tend to obey that type of size distribution. And then there's a nice one by Risevich, which is kind of a two exponents, a sum of 2 exponential. There's an old one from Carter, which was a single exponential. And they found it fits data better than, and of course there's more free parameters. So the more free parameter you have, it's just like any polynomial, you can fit better data. So just so you know, there exist a whole bunch of them. And these are the differential size distributions in number per meter cubed per micrometer of bin size. As we say, the optical regime are primarily determined by size and have to do with the size to the wavelength ratio. Not only that, it's also size to, so this row, so there's the size to the wavelength, and then there's size to the wavelength time n to the -1. So change in index refraction between the particle and the medium, because that determines the phase shift between the wave that goes through the particle and the wave that goes outside of the particle, and therefore the interaction between them as they cross the particle. So we're moving, again, you've seen it before, from dissolved organic material all the way to bubbles. And then geometric optics is where we are. And we mostly are in these two regimes. Phytoplankton, at least. Phytoplankton and sediments are in these regimes. Zooplankton. And then here it's more viruses, colloids, the stuff that goes through a 0.2 micron filter. Okay, so what happened? Just a few results that you'll encounter that have to do with particle. And again, some of it is going back through what you've seen already. We talked yesterday about cross sections. Cross section for attenuation, absorption, and scattering. They're the geometric cross section, pi d square if you're a sphere. Otherwise, it might be the cross section over all orientation. It's going to be an average g times this efficiency factor that has no units. This is the optical cross section. This efficiency factor have analytical solutions in, and we'll encounter them tomorrow in the lab, for a variety of these regimes. For the Rayleigh regime, it's this. This is the image, the imaginary part of the complex index of refraction. In a second, we'll describe it. This complex index of refraction is your real part, which has to do with the ratio of velocity, light velocity in the particle versus the medium, and then there's this imaginary part n prime, which has to do with the absorption properties of the particle. Okay, we'll get there in a second. It has to do with the absorption as you go of the material within the particle. And again, it goes like lambda to the minus 4, as you just seen in the previous lecture. So this is for particle very small relative to the wavelength, Rayleigh regime. When you're very large, at the other end, your cross-section we know is 2. If you even have some small absorption, cross-section for not cross-section, efficiency factor. The cross-section is twice the optical cross-section, once for scattering, once for absorption. That's very easy. It makes our life very easy in terms of what should we expect when we have a culture with a certain number of particles that are significantly larger than the wavelength. And there's some analytical solution for part of the VSF in this regime. So this leads to a dependence. So if you see this, do you see the wavelength anywhere here? Is the wavelength appearing anywhere here in these Qs? No. So we expect things to be flat. Okay, so scattering is going to be flat. Absorption is going to be flat because we absorb everything that goes through. And attenuation is going to be flat. So this is the difference between a lambda to the minus 4 here. So we have a lambda to the minus 4 here, and then we're going to flat. So somewhere in between we have to bridge. So here is an example of how you'll bridge in terms of QA. So that's the efficiency factor for absorption. You're going to start low, it's going to increase a lot, and then we'll asymptote to 1, to the value of 1. If you look at scattering, it's going to go up. Remember, it was very spectrally dependent, and then it's going to oscillate and going to go to eventually 1. This is 2 it starts with, but once absorption goes to 1, QB falls down. And if you look at this guy, the light absorbed or scattered or attenuated for one particle in a meter cube divided by its volume, actually it's very efficient absorber when it's small, because every photon that goes in, there's no shading. And then you're getting this, it's just another way to look at packaging, the loss of light as you go. Now, the interaction here, as the index of refraction changed from 1.02 to 1.2, is due to the fact that you have more bounces within the particle, so more opportunities to get absorbed within it. And this is what I showed you also before. This is just scattering. This is the volume-specific scattering coefficient with the resonance that changes as function of index of refraction. So just useful for us to get a sense of what to expect if I'm small, if I'm big, and what in between for these. And we can do it with instruments. So here is data from a paper where they had a scattering sensor, and they just ran different amounts of beads of known numbers. And all they did is normalize. This is in turbidity unit, so NTU. Those of you who know about turbidity. But it's proportional to scattering, divided by milligram per liter of the beads they had. How many of them they had, or how much mass of it they have. And you see exactly this resonant curve. So there's a certain size, in this case 1 micron, where they got the most bang for the buck. This is what I told you yesterday. There's certain sizes where we're getting more interaction with light than others if we normalize by the mass, by the volume. And they change depending on the index of refraction for a given, this is in micron, it says millimeter, it should be micron. And it's different if you look at the Mie solution for backscattering. Backscattering, you get more for smaller particle, 0.1 micron particle. And this is primarily the reason that we have this thing, we haven't mentioned it yet, that was called the backscattering enigma. The backscattering enigma, it's this long-lasting debate in the optics community where the signal we measure from space, if we use Mie theory, we just couldn't add up enough to get the backscattering that we measure. And so people thought, oh, it's because most of the backscattering is submicron particle, and nobody is measuring their size distribution. So it might be these guys that are the reason. It was Morel and Hahn and a variety, and Stramsky, a variety of people said, this is the reason. And then other people said, no, 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 no. It's simply because me theory does not apply, particularly in the back direction, when you deal with particles that are not homogeneous spheres. And I'll show you some things that have to do with it next, so some results. But before we get to there, one of the big difference in terms of VSF, as we talked about, or direction of scattering, is a change as function of size. Small one, Rayleigh scattering, me scattering, me encompass this intermediate range, but me can really have all the sizes in it. The me solution has all of them. You get more scattering to forward direction. Light is coming from this direction. And then finally, where you're very large particle geometric optics, most of the light goes in the near forward. And you've seen these already. This is with the list. The list is used. It's sensitive to size because it's measuring the VSF in the near forward direction. And as Colin told you yesterday, it's much more sensitive to size, here described by the slope of the size distribution, than it is to index of refraction. So less to composition, more to size in the diffraction limit of particle. So near forward scattering, strongly passing on size, less on end. So that's why we use this to get information about size. Another things we use to get information about size, which I mentioned to you yesterday, has to do with the spectrum of beam attenuation. Spectrum of beam attenuation is size dependent because the maxima for a given type of particle changes position as function of wavelength. It's easy nowadays to measure this. You measure it with an AC meter like you did in the lab yesterday, and you can do it by even do it in calibration independent mode. You measure total particle, looking at the spectra, remove the dissolved part, and the difference, calibration independent, and I just fit a power law to it. And here are data from Barnard et al., so where you see that it's changing in the ocean. This is from an old paper of mine, and this is a compilation of 40,000 spectra from Tara. And you can see that, yes, it changes. And it's well approximated, except near 676, by a power law like, so you don't see all the absorption peaks and valleys because it's beam attenuation. And then back to Volt's theory that I showed you yesterday, if you have a power law size distribution, there's a relationship between this and the spectrum of beam attenuation. If it's me, meaning they're spheres, they go from 0 to infinity, and they're non-absorbing. So we expect a relationship. So the first place we went to test this is in a bottom boundary layer. Why did we go there? Because resuspension is a good thing that's going to differentiate big particles from small particles. I mean, this is why this was a good place to test this. We went there because we got funded to go on a cruise. Science is often done very differently than the way we describe it in papers. In papers, we say, oh, we wanted to study the size distribution. We went on a boat. We measure it in the bottom boundary layer because this is where it's the best, easiest type of particles. And we show the result. But that was never how science is done. Science is done. You get a postdoc. I got hired as a postdoc in Oregon State. I was thrown a data set and said, try and find some relationship between whatever and whatever. See if you can see something that works. I'm not joking. This is how we do science. But it's never how we report it. So I get to Oregon State. I get this data set. And lo and behold, we look at the bottom boundary layer and we see big changes in the spectrum of beam attenuation and big changes in concentration because of resuspension. So what happens in bottom boundary layer? We have shear. Shear because there's a thing called the no slip condition. Right at the boundary at the bottom, water has to stop. No matter what, it cannot slip. It has to stop. So that forms a stress on the bottom. And that stress, if sufficiently strong, starts throwing particle up the water column. The big particles settle fast. They settle, as I told you, like diameter squares. So have a settling tube. So the big particles settle fast, and you'll find them deeper down. And at the top, you have the finest particle. Even if they're made of the exactly same material, they just don't settle. So here it is when it's fully settled, almost fully settled. There's still some turbid stuff. Now there's a storm passing by. And you'll see what's happening in here in a second. You see the biggest one are down below, and then slowly, slowly, this is going to clear. And so it's clearing slowly because the big particle fall really fast, and then smaller particle and smaller and smaller are falling down. So this change, there's a change in concentration, more concentration near the bottom, and also more big particle near the bottom. So that's a good place to test if what you're observing in your beam attenuation spectra is consistent with size. Maybe it's just a theory from the atmosphere. It doesn't apply to the ocean because the size distribution is never really a power law or something like that. So we go, and so I get the data, I look at it, bottom boundary layer is here. You can see it's mixed in temperature or density. Here it's density. Then you look at concentration. Indeed, concentration increases towards the bottom. This is 1 day after hurricane passage. Colin was on the boat. She can tell you how much fun it was. And then this is the change in this exponent of the beam attenuation. So when it's big, it's small particle. When it's small, it's larger particles. So we get stratification as function of size and as well as concentration. So these are bigger, smaller, small particle, big particle, small particle, big particle, small particle, big particle. Yes! I have a paper. So then, but you cannot send it to reviews because it said, okay, but you didn't show us any information about size. So thank God somebody else was on the boat, and all he did was measure size. This is Gardner. Wilf Gardner, he was looking at particle, measuring size with his Coulter counter, and so we matched those to the spectras we had. You have many more spectra with an optical instrument because you can be there all the time. It's measured at 6 hertz. You do some binning, but still, as you know, it's much harder to take individual samples. And what we're finding that indeed when there's more in the bottom boundary layer, when there's more beam attenuation, there's more particle, and they tend to be flatter size distribution. And when there's less, they tend to be on the smaller size. And the difference is not exactly 3 as theory would predict. It's somewhat 2.5-ish, something like this. And these are data from a day after the hurricane, and they are completely outside this curve. And we're still trying to understand why. We think it has to do with breakage of aggregates, and then aggregation bringing you in this direction. So we saw them consistent, about 30% of what theory tells you, and support the use of gamma, this beam attenuation spectrum parameter, as a tool to estimate size distribution slope, if you have a power law size distribution. So then a lot of people are really excited about trying to do the same with backscattering. Why? Because backscattering, our signal from space, depends is proportional to backscattering. It's not proportional to the beam attenuation. So if backscattering also has a spectrum like this, a power law spectrum, then maybe we can do it. So with a student that took the class, was a TA in this class, Wayne Slade, we analyzed a whole sheet of data from, this is 2 experiments south of Martha's Vineyard. We had a tripod in a bottom boundary layer. Again, we don't want to deal with, if we can, with phytoplankton. They're much harder to deal with. We didn't want the absorption feature that are decaying our backscattering. We wanted this to be a decent fit. We looked at that, and indeed we find backscattering spectral slope changing in a way that's consistent with a change of the least reported. You're going to use the least today, so the near forward scattering size. And this is a whole bunch of Monte Carlo simulation propagating all the possible errors we could think of, uncertainties. Here we do the same as we did in the previous paper with attenuation, and what we found is that for the attenuation, it works, but it works much better. We don't have this group of, if we only look at particle that are smaller than 20 micron. Reason is we're mostly sensitive because of acceptance angle issue to those particles. because we have a 0.93 acceptance angle. We filter out, basically the AC 9 meter filters out the effect of the larger particles. And if we keep them in, we can describe well with the slope the change in particle size distribution. So that was a pretty cool result. But so far I have not seen any paper that really is able to link this to a size distribution near the surface, which is the holy grail. We really want to be able, for remote sensing, to get size. And the question is also, is this a reasonable model? If you have absorbing particle, this is not going to fit well in regions where you have strong absorption peak and absorption valleys. But it's the default model in all the inversion algorithms. So let's hope you guys can come up with a better way. It's a basis of many PFT inversions. Another method by a person who was a TA in this class and was a student in this class. And this is Nathan Briggs. So what he looked at is the noise in the data. So there's a theory going back to Schifrin that's called the method of fluctuation, that if you have rare large particle in a suspension, you'll measure them as spikes in your relatively rare compared to the rest of them, as spikes in your signal, because they come in and go out. And the intensity and the number of those spikes, so if you look at the variant to mean ratio of, say, beam attenuation or another optical property, and then you have sample volume in it, you need this QC, the efficiency factor, and something about the measurement, how long the sample is in your field of view, you can get an estimate of the average size of these particles. And indeed, he was able to do it in the lab. We did an aggregation experiment where we aggregated, we took clay, we put in a solution where there's salts, and what clay does when there's salts on, they start to make big particles. And we had the list in there, and we had the AC meter in there, and we had open on the seaside, we had acoustics, all bunch of things, all of them trying to see this, how properties change as this stuff aggregate and get bigger. And what we find is this method of fluctuation worked nice. This is log 10 in comparison to the list. The list was our validation for size. But this he did with an AC9, he did it with the C side of the list, and he did it with a backscattering sensor. All of them, the fluctuation in them, were related to size, these statistics of fluctuation. And so far, this method has not been widely used, other than him. He uses it on Argo floats, he uses it on many platforms, but very few people have picked up on it. So to summarize, size matters, but it needs to be defined. Given that it varies order of magnitude, using the diameter of an equivalent sphere may be reasonable as a first order approximation. So the first order, it's not too bad to say I'm going to assume a spherical sphere because I have really big particle and really small one. You've seen in Ivana's lectures, the whole range of phytoplankton. But there's second order issues that have to do with non-sphericities that you have to think about. All data looks great on log-log plot. So if you really want to test your data, try and not put it on log-log plot. The calculus of size distribution is not very complicated, but the details are important, particularly unit, as were mentioned. And optical techniques, in general, are useful to constrain size. So if you're interested in size, you have in your hand tools that can help you. Can help you do it, but there's still a lot of work to be done, particularly with this. This, I feel, is ripe for work. A lot of work on. And in this class, we have two spectral beam attenuation meter. We have a BB9 for you guys, and we have a Hydroscat 6 that you guys will be using. And we have the list. So we cannot deploy the Hydroscat in the lab, but you guys are going to be measuring stuff at sea, and you can see whether you can start picking up this kind of information. Okay, any questions? Yes?
Way back to being Terra, what were the numbers?
The what?
There were numbers, the Terra thing, there were three lines.
Sorry, the 3 lines had to do with the spectrum for when chlorophyll is less than 0.1, when it's between 1 and 0.1, and this is chlorophyll bigger than 1.
Okay.
Sorry. So these numbers are how many spectra represented this. So obviously it's mostly open ocean data. Thank you for asking. So this is kind of a compilation of everything and this is from only the first 2 years of Tara. Now we have probably twice as much data from other expedition where we basically have the ACS continuously doing measurement filtered, unfiltered, filtered, unfiltered as it travels the world. Yes?
So this gamma
Yes.
that we're talking a lot about, this is the
fit. This is, you go and fit to this curve. You take this curve or the curve you got in the lab, not a curve for cultures. So this is something, if you are dealing with a mono-specific harmful algal bloom, or you're dealing with a culture in your lab, you don't expect your curve to look like this. Because you're focused on a very small part of here, of this curve. You might have, all your particles might be here, or here, or here, and you can have any kind of spectral slope. It's only when you have a wide size distribution with a lot of particles from many different sizes that you expect it, that you expect something like this. And then you go and you fit to the spectra you measure, which you'll do today. You fit a function, you say, okay, I believe that maybe that CP lambda, so the particular beam attenuation or my backscattering spectra, whatever it is you're measuring, you're trying to see if it fits a power law function of this sort. And this is the size parameter we're looking for.
right, and so that comes from something after me theory.
So this comes from me theory. So again, just like you heard about Fournier, this is Volz doing a calculation in 1954. It's reported in the book that you have in the back there. This is in German. I cannot read German. But this is reported in Van der Halst.
So that's
and that's this. So you
to me theory.
This is a result based on me theory for a specific kind of particle. A
On
non-absorbing,
me theory.
that has a size distribution going from 0 to infinity,
Okay.
and that the size distribution has to be power law. If that's the case, and they're all spherical and homogeneous, then you expect this, and this is related to the power law of the size distribution.
Of the actual.
This is theory.
Right.
And the question is, does it work in the real world? And what we're finding is it's not that bad. Even though we don't have particle going from 0,
Yeah. it's a lot
and
of
definitely
stuff.
not to infinity.
Yeah.
I mean, you did the paper too, with it, where you truncate the size distribution, because as you saw earlier,
Most of the
the important part of the size distribution for beam attenuation from the visible is
yeah, so
the micron size.
most of the contribution is going to come from these particles. So as long as this assumption is good for these, you probably will find a relationship.
Right.
That's what Ken is saying. As long as, below 10 and say above half a micron, you get a power law to fit relatively well. Even though they're absorbing, and the theory is for non-absorbing, it seems to work.
So it starts with me theory.
Yes,
Volts
Volts 9054.
comes up with.
And he does it for aerosols. I mean, this is the other beautiful things in optics,
get
is
a
you can take
particle.
from astronomy or from atmospheric science result and try and see if they work for the ocean.
Any true particle distribution and then relate it to beam attenuation.
Yes.
Through gamma.
Yes.
And that's sort of like what people are interested in, is this gamma.
No, people are interested in this. You want to know size.
Right.
But you don't measure size everywhere. You don't have your Coulter counter, or you don't have, and it's difficult, and so you're trying to get at least some description of the tendency of size.
Through beam attenuation. Right.
Because this is
wanna
easy to
back
measure.
out
This is
the-
easy to measure. This is hard to measure.
Right,
Yeah.
right. Okay, I see. Yeah, you start with that and back out
Yes,
to the size
exactly.
distribution because that's.
And this is only one parameter. With the list, you get many more degrees of freedom out of 32 bins, maybe 8 degrees of freedom about the size distribution. Here you get one indicator of size distribution.
Can
Yes.
you explain the fluctuations a little more?
I strongly recommend you read that paper, but it has to do with the variance to mean ratio.
Of these?
of the optical parameter you measure. So you measure a variance in beam attenuation or in backscattering. Here it's backscattering. Here it's beam attenuation of one instrument and here of another. You need to know something about the efficiency, but if they're big, this is not too far from 2. The volume you're imaging, and this will give you something that has to do with the average cross-section area of your dominating particle, and that will come into, and you'll back out by the square root of it, what's your diameter.
So there's more measurement variation in the beam attenuation of large particles, is that?
Yes, yes, because they're bigger, because they're bigger.
That's really cool.
It is really cool. What?
It has to do with the beam, right? So if you look at the cross-section of the beam and you have small particles, there's so many small particles in there that if they move in and out, you don't see big fluctuations, but if a particle moves in and out, you see it. So it just has to do with when you look at the noisiness of the time series, you can say something about whether it's big or small particles, and then you actually got to the point where you can quantify it. It's really cool.
We haven't shown you any time series data or profile data with that resolution.
I showed only profile data yesterday, but nothing about fluctuations. But you'll see it in your own data. And you see it in your lab data. In your lab data, you have fluctuation. Actually, you can try and see if.
I think I'll probably show it tomorrow stuff that he tried to do on using the flow data in North Atlantic with the same approach and trying to kind of differentiate the size of the particle, the size of the phytoplankton using the chlorophyll fluctuation, and then estimating the aggregation of the particles that pertains to the export and scattering.
So this is really completely underutilized as a method. There's tons to do.
Learn from Nathan, publish your stuff, because it's not published yet. So at this point, you know what? Publish it yourself. Okay, so let's start this. This was done 2013. I did this plot, I'm going to show you that the same year. What is now? 2019. Can you take this off the It's going to kill me.
I go. Any questions? Other questions?
This method's mainly for looking for big particles.
No, it's for looking at the dominating particle in your suspension.
Dominating
Yes.
particle.
So those either that there's a big concentration of, or yes, or large, dominate in terms of cross sectional area.
Okay.
Yeah. So it's cross section area weighed on your size distribution.
That's cool.
Yeah. And you can do it with gliders. You can do it with
from just beam attenuation.
backscattering, and these are beam attenuation. Any optical variable that you're measuring. He did it also with chlorophyll fluorescence.
Yeah. So that's backscattering at one.
At one wavelength, just like you have on your gliders.
Yeah.
Other questions? So there's a group yesterday that did not finish the chlorophyll stuff. That's what you're going to start by doing. The other 3 groups in about 15 minutes. 1 will be here, and 2 will be in the lab. Okay? In the Mitchell lab. And what you're doing today is you're going to measure scattering with 1 instrument, backscattering with 2 instruments, attenuation with 2 instruments, near forward scattering with 1 instrument. You're going to measure turbidity, and the way it's going to be, each group of you is going to have a sample. There'll be a group that's going to be the sediment group. They'll have the Arizona dust, slurry, suspension. There'll be 2 groups with phytoplankton cultures, and then there'll be a group of the Damariscotta River estuary. And so you'll get to see kind of the contrast between these difference in terms of backscattering ratio, how does the beam attenuation look, all kind of stuff that we've been talking about, but now you guys can calculate. Size distribution based on the list. And we have 2 list new experts, which are Juliana and Kuhn. Juliana is hiding. They've been playing with the list for a while, so they have already software for you to back up once you get data to play with and get both the shape of the volume scattering function and okay?
Cool.
And I'm sure you have list software too. Limited, okay.